Hello, my students. So we're going to continue our discussion of cell injury. Remember, in the first two videos on cell injury, we primarily focused on hypoxia and respiratory burst or oxidative stress, and it's all of its consequences. And now we are going to take a look at how cells can adapt to cell injury. So remember how we started out our discussion of cell injury by saying cell injury can be reversible or irreversible. So when we studied hypoxia in the two previous videos, we learned that hypoxia can be reversible to some extent, but also irreversible and lead to cell death. So in this video where we do focus on cell adaptation, we're looking at reversible types of injury. So it's kind of amazing at how well our cells are capable of dealing with all kinds of injuries. You know, just think of the things we do with our body, and therefore all the things our poor cells are exposed to, whether it is what we eat, what we inhale, what we swim in. I mean, what we take as medication or not. Um, it's just endless, all the kinds of threats that we expose ourselves to. So cells are do well in surviving and preventing dying. Now they do die sometimes on purpose because they're programmed to die, and so we will discuss that as well in another video. But when cells are exposed to what we call an appropriate stimulus, meaning a stimulus that's not going to kill them, we say that they are adapting. And these adaptations can usually be reversed after the appropriate stimulus stops. So the kinds of adjustments that cells make are listed here. So we might see that literally they change shape, the cells. So their morphology, morph being the shape of the cell changes, or they might start accumulating all kinds of things in the cells in an attempt to deal with the types of injuries that are affecting them. Or we may even see that organelles go through changes in an attempt to deal with the somewhat harmful, well, maybe we should say harmful stimulus, but a stimulus that forces them to make some adjustments. Let's put it that way. So earlier I said that cells can adapt by changing their morphology, and morphology does refer to shape. And indeed, they can change their shape by changing their size. They can even change cell type. And we're going to add to that as well how they can change the amount of cells. So when it comes to terminology in cellular adaptations, you're going to see two Greek roots. One is called trophy, and that refers to nourish, while the other one is plasia, which refers to form. I will be honest, these translations don't really make a whole lot of sense. So you're going to have to kind of memorize in English really what they truly mean. And so when it comes to trophy, we're typically going to see that the cells are either going to grow bigger or smaller, right? While plasia refers to the cells increasing in number, or they're even switching to a different kind of cell. So here we go. So atrophy means that we are without growth, essentially. So the cells are going to grow smaller, while hypertrophy is the opposite. You know, if you in AP1, you've learned that, for instance, during embryological development of skeletal of bones, for instance, where we start with hyaline cartilage, cartilage cells get bigger and bigger and bigger. So we say they hypertrophy. Or when you learned about skeletal muscles, you learned that the reason why we bulk up is not because we make more muscle cells, it's because our skeletal muscles just increase in size. So that is what we call hypertrophy. And then we have hyperplasia, which literally means that we make more cells. Metaplasia means meta meaning middle. We're going to form different cell types, um, as in we're going to go from one cell type to another cell type, right? So we're not really making more cells. We're not losing the size of our cell. We're going to just stay within the middle plane, I'll just say meta, and go from one cell type to another. And then dysplasia, anytime you see the term this, it always means that there's some irregularity of sorts. So we're forming um, abnormally looking cells, usually a precursor to cancer. So, let's say that you have always been very active working out while you were in high school, and now you are taking college courses, you're working full time, you got married, have got some kids, really very little time to work out. And so you might have lost a lot of mass in your tissues in your muscle tissues, and that's what we call atrophy. Literally, a lot of the proteins, those thick and thin filaments, are broken down and resorbed by the cells because they're really just all amino acids once those proteins are broken down. So that example I just gave you of not using your skeletal muscles as much, so you're not as bulked up as much, is in a form of what we call disuse atrophy. You're literally not using the skeletal muscles as much, so you lose a lot of skeletal of muscle mass. But people, for instance, who become paralyzed are also going to lose a lot of muscle mass because their nerve supply is damaged to the skeletal muscles due to a spinal cord injury, for instance, or it could even be due to a very damaged nerve. We might even see ischemic atrophy. Remember, ischemia means low blood flow, um, which can result in a stroke. And then there's something called physiological atrophy, which we'll take a closer look at in the next slide. So for instance, you may lose a lot of muscle mass when you just don't have enough nutrition, right? You might be very active. You might be, uh, gosh, walking all day long trying to flee from an area. I don't know. But if you don't have enough nutrition, you cannot build up enough protein to build up those muscles. And so you're going to see that you're having a form of physiological atrophy. But physiological atrophy is also very often due to changes in hormone levels. And I think the best thing that many of us can relate to is when women go through menopause. When they go through menopause, their estrogen levels drop significantly. I mean, estrogen levels drop in men as well, but they really drop significantly in females. And this has all kinds of consequences, particularly when it comes to the reproductive structures. We're going to see that there's going to be some thinning of the smooth muscle wall of the vagina and the skeletal muscle wall. We're going to see that the breasts are going to diminish in size. So those are forms of physiological atrophy. As we age, our thymus pretty much disappear. In old people, there's really not much of a thymus left, and this all has to do with how our immune system functions, particularly as our adaptive immune system. Um, as we age, it just becomes much more difficult, for instance, to build muscle mass. As we age, entropy takes over, chaos takes over in the body. Things just don't work as well. And so it's a natural aging process. Now, we also see physiological atrophy as a consequence of abusing um, hormones or prolonged use of hormones. So, for instance, um, men who abuse steroids, for instance, here it says androgen abuse, basically steroids, might see that there's a significant change or decrease in the size of their testicles, right? What's the point of their testes of cranking out all this testosterone if they're taking the supplements, right? There's no point for his own body to keep secreting that particular steroid called testosterone. People who are for a long time on prednisone might, to as will learn when we study inflammation and disorders in the immune system, prednisone is going to reduce inflammation, is going to be acting as an immunosuppressant, often taken by people who suffer from autoimmune diseases. Um, and prednisone is a type of steroid, a glucocorticoid to be more specific, that is produced by your adrenal gland. So we might see that in those particular patients, their adrenal glands begin to decrease in size. Hypertrophy is the total opposite of atrophy, where we see that normal cells here are now going to get bigger and bigger and bigger. So hypertrophy is not the same thing as hyperplasia. In hyperplasia, if these are our normal cells are going to just make more of themselves. So let's, for this slide, focus on hypertrophy, where the cells get bigger and bigger. I gave you examples earlier already, skeletal muscles, hypertrophy, but so do our heart muscle, or our heart muscle. If in many athletes, I'm talking about professional athletes, they might have a hypertrophied heart. Again, just like in skeletal muscles, it's not because more cells are made. Nope, we're not discussing hyperplasia, we're discussing hypertrophy. So literally more and more and more proteins are synthesized in these cells so that they grow bigger and bigger and bigger. So what are some triggers for hypertrophy? What are some causes of hypertrophy? Well, we already talked about using muscles, right? So stretching them, literally using them, is going to trigger them to hypertrophy. But even for many cells, being stressed by not having enough ATP might do it. Activation of cell degradation products. We'll talk more about those hormonal factors and trying to compensate. So let's take a look at some of these things. So we've already talked about how exercising, working your skeletal muscles and your heart is going to lead to hypertrophy. So we're now looking at examples of physiological hypertrophy. Clearly, this guy is doing a lot of stretching of his muscles and working his muscles so that they are all hypertrophied. We can also see hypertrophying of adipocytes, meaning that they collect, or they start to create more and more triglycerides inside of them. And this is typically because we eat too much or beyond the amount of calories we really need. So in addition to physiological hypertrophy, there is also pathological hypertrophy, and we have two forms, adaptive and compensatory. Let's take a look at the adaptive type of pathological hypertrophy. And I think we should start with our second example here. So a person who suffers from hypertension, persistent increased blood pressure, chronic high blood pressure, that person's heart is going to have to work harder and harder to push the blood out of the heart into those vessels with their high blood pressure. And so ultimately, that is going to trigger those heart muscle cells in the ventricles to grow bigger and bigger. And so we see a thickening of the ventricular wall, or hypertrophy. Another example would be that smooth muscles get thicker because there has been some kind of obstruction, perhaps a prostate has been obstructing the flow of urine in a man. Compensatory hypertrophy is quite common when a person, let's say I have a friend who donated one of her kidneys to a friend who needed a new kidney, and so more than likely what's happened is that her remaining kidney has increased significantly to compensate for the loss of the other kidney. So these are two examples of a pathologic hypertrophy. And remember, in the previous slide, we looked at physiological hypertrophy. Hyperplasia refers to us seeing an increase in the number of cells. So if we have 4 cells, we now see when cells are going through hyperplasia, 1, 2, 3, 4, 5, 6, 6 cells, let's say. And this is going to be, if it's adaptive, it's going to be a very controlled thing. So we're not per se looking at cancer here. The other thing to keep in mind is, in order for cells to be able to make more of themselves, they have to be able to go through mitotic divisions. Now, skeletal muscles cannot do that, heart muscle cells cannot do that, and neither can neurons. So that leaves other cells that have the capability of doing this. So in the case of physiologic hyperplasia, we again see that we have hormones that can trigger hyperplasia, as you see in a woman who becomes pregnant, right? Her breasts increase, her uterus increases due to the changes in hormones. Or we can see compensatory hyperplasia, a person who is lost or had to have part of his or her liver removed, we'll see that that liver can actually grow back to some extent um, due to this compensatory mechanism that is part of physiological hyperplasia, literally making more liver cells. And just your ordinary type of wound healing is a normal process of hyperplasia, therefore physiologic hyperplasia. Now, we saw pathological forms of hypertrophy, and we see pathological forms of hyperplasia. And so in this case, we see hyperplasia occurring in response to abnormal hormonal stimuli. So it could be that a person, of a woman, is producing excess estrogen to where we're now starting to see that the inner lining of her uterus becomes overly thick, leading to um, uh, you know heavy bleeding. That would be an example of a pathologic form of hyperplasia. Metaplasia means that we're going to see that one cell type is replaced by another cell type. We often see this happening in the case of epithelial tissues, where we see one tissue category, um, let's say the pseudostratified ciliated columnar epithelial tissue in the trachea is replaced with stratified epithelial tissue. And usually this is pretty controlled and is somewhat reversible, but a person who continues to smoke for years and years and years and years is not going to be able to reverse this anymore after a while, and could potentially end up with cancer. So here we see an image or a photo illustrating what I was talking about, how pseudostratified ciliated columnar epithelial tissue that is illustrated here in the trachea. You can't see the cilia. You're going to have to assume they're there, but you do see the goblet cells. How smoking can cause or trigger these cells to adapt and become um, a stratified squamous epithelial tissue, which of course is more protective to the underlying tissues. Another example would be in the esophagus, where the stratified squamous epithelial tissue is actually replaced by simple columnar epithelial tissue due to gastroesophageal reflux. You may have heard of GERD. When does this happen? In the trachea, particularly in response to smoking. In the case of GERD, it means that a person is often seeing some of the stomach acids move up into the esophagus, constantly irritating the esophagus and therefore causing lots of inflammation. So chronic irritation with inflammation can cause metaplasia. That leads us then to dysplasia. And remember, anytime you see the prefix dys, it's implying something irregular. You might hear the term dysrhythmia, referring to irregular heart beating. And so here now, in the case of dysplasia, we're looking at cells that are starting to look very irregular. meaning their size is different, their shape, they just look all different. It all looks rather abnormal. Um, and despite the fact that dysplasia is really an abnormal look for cells and considered adaptive, it can, these cells supposedly can still reverse to normal after removal of the stimulus on time, obviously. If, on the other hand, this is not, the stimulus is not removed, or this is not controlled in a decent time period, it could result in cancer. So we women are pretty familiar with the term dysplasia because we often, we have to go see the gynecologist regularly to have our cervix checked, which is basically the transition area between the vagina and the uterus, um, where cells are collected via what we call a pap smear. I'm hoping all of us girls are doing that, named after Papa Nicolau, in order to make sure that the cervix cells look healthy. And clearly here, this person, an asymptomatic patient, went in for a regular yearly pap smear, and clearly we see that there are irregular looking cells. Here are the normal looking cells. So very different looking cells there. What triggers this? Well, um, often it is as a result of constant problems due to sexually transmitted diseases, but it could also be that a person is quite allergic to contraceptives, a variety of other things. And then also exposure to substances that cause birth defects can also trigger dysplasia. Okay, so let's do a quick reflection on what we've done so far and what we're still needing to do. So far we've looked at how cells can somewhat adapt to appropriate stimuli by changing their cell morphology. So they can change their size and shape somewhat. They can change the number of cells, and they can change, what's the last thing, the cell type, right? Maybe go from a columnar cell to a squamous cell. So we still need to take a look at intracellular accumulations and organelle changes. So when cells are being injured, they try anything and everything to survive. We looked at how they can change their morphology, but sometimes they end up accumulating all kinds of stuff inside and don't have the proper mechanisms to get rid of whatever they've collected. I said stuff, but a cell might end up, for instance, with too much water, or too many proteins, sometimes even pigments, too much lipids, etc., etc. So we're going to take a look at the mechanisms that create these excess substances inside of cells. We're going to take a look at abnormal metabolism. We're going to look at protein problems that involve problems with this structure called a proteasome. We might not have the right deficits. We might not have the right enzymes, I'm sorry, or there might be just some things that accumulate in the cells that are just not possible for cells to digest or get rid of at all. So it might not be a matter that they accumulate in large amounts, it's that the cells just don't have the mechanism to dispose of them. Now, I purposely use numbers here, like this, because your course objectives will ask for these four, so that you can follow your course objectives better. So sometimes cells just cannot metabolize something properly to where it then accumulates. And when we studied hypoxia, we learned about hydropic swelling, when the sodium potassium pumps can't function properly, sodium accumulates in cells, and water follows. So this is a form of water accumulation, too much water inside of a cell. That's a form of an intracellular accumulation. Let's take a look at lipid accumulation and carbohydrate accumulations. So recall that there are different kinds of lipids, and the neutral fats are often referred to as the triglycerides. These are the ones that get stored in our adipocytes, right? So triglycerides are also considered molecules that literally store our fatty acids, right? Fatty acids together with glycerol form triglycerides, and we depend on these triglycerides a lot for energy. Now, people who are alcoholics in particular, but even people who suffer from some form of malnutrition, whether it is overeating or not eating enough, in addition to people who suffer from diabetes mellitus, might end up with problems with their liver to where they accumulate too many triglycerides. And we see this a lot in alcoholism, and there it is typically referred to as fatty liver. And so here, for instance, you see how cirrhosis in an alcoholic forms. Here we have a healthy liver, but all of these triglycerides are going to start piling up in the liver because the liver cells are getting damaged due to the excess intake of alcohol and possibly drugs as well. The damaged hepatocytes are the liver cells, are going to ultimately start getting replaced with scar tissue. Yes, liver cells are epithelial cells, they can divide, but there comes a point where they just cannot keep up with replacing themselves, and we're going to see that this scar tissue forms. We call that liver fibrosis, and if that continues, we're going to see a cirrhotic liver that becomes literally non-functional, and the person will ultimately die if they're not careful. Cells can also accumulate the polysaccharide called glycogen. Remember, glycogen is basically the storage molecule for glucose. So if you stick a bunch of glucose molecules together, I'm going to put a little N here to imply that we have a long string of glucose molecules, then we form glycogen. And muscle cells store glycogens, and the liver stores glycogen, and when we're running low on glucose, for instance, our cells, or I should say, the proper hormones are released, I should say, to where glycogen is broken down in these different organs or tissues so that glucose can be dumped into the bloodstream so that it can be used for aerobic respiration and make ATP. But, we have diseases out there to where a person may end up with too much glucose, as is the case in diabetes mellitus. Of course, you'll learn a lot more about learn more about diabetes mellitus in pathophysiology too. It might also be that literally a person does not have the right enzymes to break down glycogen into individual um, glucose molecules. And this is typically an inherited condition to where that person is lacking that enzyme. So they're missing the gene that codes for the protein, namely the enzyme that can break down glycogen into individual glucose molecules. Um, Aside from those cells I mentioned, the liver, the muscle cells, also the nerve cells and the kidney tubules can accumulate quite a bit of glycogen because of the issues that we discussed here. So we just finished discussing all these abnormal metabolic processes of intracellular accumulation. So let's take a look at what's next on our list. So now it's time to take a look at the various protein problems that might arise due to malfunctioning proteasomes or we're missing an enzyme. So let's take a look at that. So in our cells, even in the nucleus of our cells, we have these interesting protein complexes called proteasomes. And they have the ability, think of them as almost a little machinery that can um, that proteins enter, they kind of enter through a little door, and now they're in this big plant where they go through from machine to machine to machine, and they're broken down into amino acids. Just use your imagination a little bit. That's kind of what a proteasome is like. And so we end up with these individual amino acids that the cell can then use to make new proteins. So it's a way to recycle possibly misfolded proteins or old proteins that need to be gotten rid of, or perhaps the proteins are somewhat damaged, right? So proteasomes are great things in that they help us reuse amino acids in proteins that can't really function anymore. But if these proteasomes are not functioning right, then clearly we're going to see that all these proteins that really normally the cell should be getting rid of um, are starting to accumulate. And for some reason, this accumulation of these proteins is referred to as hyaline at times. I'm not 100% sure where that term comes from because we also have something called hyaline cartilage. So I don't know if that has anything to do with that. So another example of a protein problem that leads to accumulations in cells is the lack of an enzyme. And we already discussed that when we discussed how glycogen accumulates. Remember that enzymes are proteins. So this is another example of malfunctioning proteins or lack of proteins and how they can then result, or how that can result in the collection of things inside of a cell that shouldn't be there. Now, if we continue our discussion on things that can accumulate inside of cells, as mentioned before, sometimes cells just do not have a way of getting rid of something, and we'll refer to these as indigestible substances. Pigments for cells, for some reason, are very difficult to digest. And when we say digest, meaning breaking them down into molecules that then the cell can reuse for something else, right? So they just sort of hang out in the cell. And we call them pigments because these are chemical substances that pick up certain wavelengths to where we can see them. They're slightly differently colored. A good example of a pigment that accumulates and shouldn't be there really is lipofuscin. So lipofuscin is a very common pigment. Many cells accumulate it, and it's definitely a consequence of the aging process. We see it as a result of damage to the cell membrane, but also especially mitochondria and even lysosomes. So in relation to mitochondria, when the mitochondria don't function properly or damaged, that means they cannot use the end products of the oxidation of fatty acids. So when fatty acids are broken down, then the end products can enter into the Krebs cycle and go through the Krebs cycle, the electron transport chain, and make ATP, right? So it doesn't always have to be glucose end products that end up entering into the Krebs cycle, proteins. Basically, all of our biomolecules, but especially proteins and lipids, can be broken down to where some of their products can then enter into the Krebs cycle and the electron and move on to the electron transport chain. But when the mitochondria are damaged, then these end products cannot be used and they end up accumulating. So we're almost done with our discussion on cell adaptations. We have one more thing to take a look at, and that is what happens to organelles at times. But let's quickly reflect on what we just finished discussing, which are um, intracellular accumulations, which is a form of a cell adaptation. So we learned how at times, because of abnormal metabolism, we might see that water accumulates or that neutral fats called triglycerides accumulate. There might be an accumulation of glycogen. As a matter of fact, the accumulation of glycogen might also be due to a protein problem because a person might have a genetic condition where they can't make the enzyme to break down glycogen into glucose. There might also be a problem with the machinery that's responsible for recycling proteins, and they're called proteasomes. And then finally, cells can collect things that they really don't have any machinery or mechanism or process for uh, to digest. Um, Substances and pigments are a very good example of that. We've only looked at one pigment because it's a common one called lipofuscin. So then let's just finish up our discussion of cell adaptations by looking at subcellular or organelle changes. So we might see that the smooth endoplasmic reticulum actually starts to get bigger um, when, as a form of a cell adaptation, mitochondria may have to adjust by increasing in number, especially when cells hypertrophy, but they may even increase in size. They may change shape. They may have to adjust their function somewhat in particularly their ATP production and calcium storage. Um, Clearly, when there are less when cells atrophy, I should say, they're going to need less mitochondria. There might also be some adjustments in the cytoskeleton where it starts to break apart. And unfortunately, then these components of the cytoskeleton might start getting in the way of the movement of some of the organelles inside of the cell, or even the cell's ability to move itself. So this was our last topic on cell adaptations. And so in summary, cell adaptations are a response to an appropriate stimulus, and cell adaptations are reversible. So they're reversible forms of cell injuries. Cells can adapt by changing their cell morphology. They can increase in size, they can decrease in size, they can increase in number, decrease in number, they can switch cell types. They might accumulate all kinds of things. Those could be things that are indigestible or just because they don't have the right enzyme to break down whatever is accumulating. They might have damage to some kind of a mechanism in the cell that normally breaks down whatever substance that is accumulating. There might be water accumulation. And then finally, we will see that some of the organelles will actually go through adjustments, particularly the mitochondria. So that wraps up our discussion of cell adaptations. So the last thing we still need to discuss as part of cell injury is cell death, which is extremely interesting. It's a pretty detailed topic, so be prepared for that.